Well, I want to thank Emily and Bob and Greg for the kind invitation to come here and present some of our recent historical information on cancer as a metabolic disease. I don't want this to be too loud. Is it too loud? Am I blowing somebody's eardrum out here or anything? No? Okay. Cancer as a mitochondrial metabolic disease is what I'll be speaking about this afternoon. And I have a couple of quotes that we'll introduce that we need to know about. I think Greg hit upon one of these in his presentation. But when we talk about, and what Dr. Palmer was speaking about, we have certain mindsets in people related to some of these quotes here from Carl Sagan. You know, we live in a society driven by science and technology, and most people have no clue what's going on. Tolstoy, the most difficult subjects can be explained to the most slow-witted man if he has not formed any idea of them already, but the simplest thing cannot be made clear to the most intelligent man if he's firmly persuaded in what he knows already without a shadow of a doubt. We have these problems. Assimilation bias or confirmation bias from Francis Bacon, the originator of the scientific method. Human understanding, when it has once adapted an opinion, draws all things else to support and agree with it. And of course, in our area, follow the money, Upton Sinclair. Difficult to get a guy or get a man to understand something when his salary depends on him not understanding it. This is really, all of these together represent some of the challenges that we have that speak to your philosophy of science in one way or another. But what we do is we document every year the American Cancer Society. One of their major functions is to log the dead, basically. New cases, deaths per year, deaths per day. And you can see it's always increasing to some degree, more new cases. And you know, we're getting almost 1,700 people a day in the United States dying from cancer. And if you do that, if you divide that number by 24, it comes out to about 70 people per hour. Okay, this is not an insignificant, this is an epidemic. We keep throwing massive monies into these problems, billions and billions of dollars, both from the federal government and from private foundations. You know, the pink ribbon campaigns, you hear all about them. People just mindlessly run out and throw money at a problem. They have no clue where the money is going. There seems to be no accountability, either in the federal government or for the private foundations. It just seems to be a year in, year out ritual of throwing money at a problem with expecting something to be different, and generally it doesn't. What I do is I check the scientific advisory boards of the individual foundations that support, and then you can know why we're not making any progress. This is a lot of words, cancer statistics from 2024. What they want to say here is that we are making major progress in reducing the death from cancer. I spoke to someone the other day, this is the most exciting time for cancer and all this. And I said, what about all the dead people? Yeah, well, that's an unfortunate problem. But one of the things that they tell us about all the progress we're making, in the early 1990s, there was this anti-smoking campaign where secondhand smoke was bothering people, right? You're sitting next to this guy, no, you can't. So it was a societal peer pressure on people that were smoking. You know, we used to have, years ago when I would go to scientific meetings, you'd have to wave the smoke away. You couldn't even see the slides, right? Scientists are all puffing away, everybody's puffing away. No, you can half the restaurant smoking, no smoking. Now if they catch you smoking, they have a team will come out and beat you half to death. They catch you smoking a cigarette. So as the result of that, we've prevented a lot of cancer deaths, okay? So the red line is all of the people that would have died had we not smoked, all right? So if we didn't stop smoking, we would have had all these dead people. But we're still on a trajectory of increase. So the biggest reduction in cancer has been a prevention one, a personal behavior one. Not what we think of all the advertisements we hear on television every night about how wonderful. Then we show you some smiling guy riding a bike, and with 15 different ways the thing is going to kill you. The problem is it's personal choice that it does a lot to reduce this. So we're going to talk about cancer. What is cancer? It's just dysregulated cell growth. People always say, what is cancer? It's cell division out of control. Is this disorder caused by genetic mutations? And we're going to talk about that. Or is it caused by chronic insufficient respiration coupled to compensatory fermentation, or what we refer to as substrate level phosphorylation? Now, a little bit talking about theories in science. Scientific theory is simply an attempt to explain the facts, the data, the facts. Reality is based on replicated facts, whereas interpretation of the facts is based on a credible theory. You collect an amount of data, and people have different ways to interpret this, and if everybody is interpreting it in a similar way, then we have a theory, we can test that. Credible theories move science forward, whereas flawed theories can stall scientific progress. Now let's look at some of these in historical perspective. The heliocentric theory could explain better the movements of celestial bodies than could the geocentric theory. Now, for 1,800 years, the ideas of Aristotle and Claudius Ptolemy were focusing on how do you explain the movements of the planets when the Earth is immovable and stable. So we see the moon, we see the sun, and we see the planets, and then we see the stars, which are actually in a separate sphere. And it was a series of mathematical epicycles, deference, where the planet is moving in one way, then all of a sudden decides to turn and go in a different way, a deference. Equants are the angle of the sun during different times of the season. It was a lot of mess for 1,800 years. And then Nicholas Copernicus up on the top decided, he struggled with all the epicycles and the mathematical problems with the celestial bodies. He said, maybe if we rearrange the organization of the celestial bodies, if we put the sun in the center and make the Earth simply another planet, maybe we can account better for the predictability of where these planets are supposed to be at the time they're supposed to be there. So again, we find predictability that was far more accurate than previously thought. Now, of course, the next guy, Johannes Kepler, understood that they were not just perfect circles, but they were an elliptical. He jumped all over. He knew exactly what Copernicus was saying. Of course, Galileo, the next guy down, developed the telescope to confirm the moons of Jupiter and also angles of the planet's movement. Was very excited. Of course, he was house arrested by the Catholic. You were threatening the power establishment at that time, which was the Catholic Church, the seat of all knowledge and predictability. Now, the poor guy on the bottom there, anybody know who that guy is? The guy on the lowest guy here. Anybody know who that guy is? Nobody? Giordano Bruno. You ever hear of Bruno? Okay. Bruno was a cleric in the Catholic Church, and he was a strong supporter of Copernicus, okay? And what happened, he ran around, told everybody that, geez, Copernicus is right. The Earth is not the center of the universe, the sun is. Well, that didn't go over well. So they stripped him naked, held him upside, and burned him alive in a plaza of Rome, okay? He's considered the martyr of science. Giordano Bruno, you should all know. Galileo got away with house arrest, but poor Bruno was burned to death. Why? He was challenging the power structure at the time. But there was one thing that Bruno said that really put the flame to the torch here. He said there might be life on other planets. Oh, that was it. That guy's gone. Forget about his ass. He's finished. And that was the end of Bruno. But the key is it started what we call the Copernican revolution, and the renaissance of science in Europe is the result of this. And then we have the germ theory, would explain better the origin of contagious diseases than could the bad air miasma theory. And the miasma theory was from Galen, a Greek Roman physician. Galen's ideas about disease permeated all the way into the 1800s. And it was Louis Pasteur who clearly showed with his experimentation that germs actually cause disease, not bad air, not miasma. So, and he was vilified by the French medical aristocracy at the time. And they said, he doesn't have an MD degree, therefore we can't believe in it. He's only a Ph.D. He turned out to be right. The MDs were not right. So and we look at this. And then, of course, there's the Darwin-Wallace theory of evolution by natural selection. Could explain better the origin of species than could the theory of special creation. And those little numbers on the top they are the descendants of original ancestors that exist. Each one of those horizontal lines is a millennial, could be millions of years, hundreds of thousands of years. He went through all this. And you can see all the extinctions that have occurred in the organization of biological plants and animals over time. I have a whole chapter in my book on cancer. Nothing in cancer biology can make sense except in the light of evolution. So we incorporate evolutionary theory into our design of therapies that we use in the lab. And thank you to Chris Palmer for at least identifying what the mitochondria are. It's actually a spaghetti network inside the cell. These diagrams that you see in textbooks make these things look like a little bean there, but they're actually a spaghetti network, and he's right, they're actually moving around, they fuse, they fizz, they have a very active participation in the energetics and vitality of that cell. So can the mitochondrial metabolic theory explain better the origin and management of cancer than can the somatic mutation theory? This is the next big transformation in medicine. Which of these theories is correct? And we're going to do a dive on this and let you, as rational thinkers, come to your own decision as to what you think might be what we're dealing with here. So does cancer arise from nuclear somatic mutations or from chronic mitochondrial damage with compensatory fermentation? There's a two-step here. You have to have the chronic damage and you have to have protracted compensatory fermentation, or what we refer to in our lab as substrate level phosphorylation. Fortunately, I have my research crew back here. If I screw up with one thing, they will attack me. So not you good folks, those guys. So, and Bob is there with the rest of them there. So I got to be careful. So anyway, we're going to talk about these concepts. So we're looking at the two organelles here, the mitochondrion and the nucleus. And the somatic mutation theory of cancer says that dysregulated cell growth is due to mutations in the nucleus that cause the cell to become growth dysregulated or growing out of control. And I will present evidence that challenges that. So we put up this paper. This is by Hanahan and Weinberg. They're down here at Robert Weinberg is at MIT. And this is one of the most highly cited papers in all of biology. It's cited by 75,000 times. So hallmarks of cancer, the next generation, a dogmatic view, an irrefutable truth, a silent assumption, if you will. Cancer is a genetic disease. And cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. Page 661 in the paper, you can read it if you have the paper. If not, I can send it to you. Okay, so this concept has now permeated all of the textbooks of biology and biochemistry, cell biology. All the medical students, when they get their first year of training and they do biochemistry or whatever, they're talking about cancer as a genetic disease driven by different kinds of mutations that we're going to look at.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
And not only that, it's also supported from the National Cancer Institute, our government cancer website, National Cancer Institute. What it says right there, cancer is a genetic disease. That is, it is caused by changes to genes that control the way our cells function, especially how they grow and divide. And then you see down on the bottom there, cancer is caused by certain changes to genes. All right, so this is confirmation bias. What it means is we all, in terms of Francis, assimilation bias, confirmation bias, the whole field says that this has to be a genetic disease because the NCI says it is and everybody else thinks it is. Not everybody, but a lot of people. So let's look at this somatic mutation theory. So if you're going to understand what the current theory of cancer is, because ultimately if you think you understand what the disease is, then you should be able to strategize therapies that manage it. And you just saw all the dead people piling up. Something's not right. Okay, so we have mutations in tumor suppressor genes and in genes or proto-oncogenes leading to cell division out of control. So we look further at this and we see tumor suppressor gene. This is a normal product of some gene, a protein of a gene in our genome, and it keeps normal cells in a growth regulated state. A mutation happens in that gene and you get dysregulated cell growth. So this is called tumor suppressor. These are tumor suppressors. But in addition to tumor suppressors, we also have these things called proto-oncogene. Proto-oncogene is a normal gene in our body. If it gets a mutation, some sort of mutation, but there's several different kinds of genetic changes. You can have a mutation in the normal proto-oncogene, and then that causes an oncogene, which makes the cells grow faster. So you can have a mutation in there, or you can have extra copies, multiple copies of this proto-oncogene leading to normal growth stimulating proteins in excess, or you can have the gene can actually move around the genome and find itself next to a different kind of a promoter, forcing the product to be in excess, leading to dysregulated cell growth. So we have the tumor suppressor genes, proto-oncogenes, and oncogenes, and these kinds of things. And this is all part of the somatic mutation theory, the current dogmatic view of what we think cancer happens to be. This is Dr. Vogelstein from Johns Hopkins, when he says that we now know precisely what causes cancer. A sequential series of alterations in well-defined, and remember this term, driver genes. Okay, not all the genes. We found out, oh my God, there's some that don't do anything, passengers, valleys, hills, all this kind of stuff. Drivers are the key. So the key to managing cancer is going to be to target the driver genes that are causing the dysregulated growth in a tumor in your body, okay, or causing a particular population of cells to become dysregulated. All these things happen. Now, here we are today, individuals' cancer cells, this genetically tested for personalized therapy. You hear about all this precision medicine, personalized therapy, all this kind of stuff. Breast cancer cells being examined to see if they possess extra copies of a particular gene. There was a company down here in Kendall Square, Boston, that was bought out for $2.4 billion. It's a company that looks at tissue from cancer patients and looks at hundreds of different kinds of mutations, and you get this nice readout. Oh, geez, look at all the mutations I have. Look at that. You know, they're looking up that, and they say, you can give you a short list for $4,000, but if you really want the Cadillac list, you can pay another $7,500, and they'll give you thousands of different mutations. And then you say, wow, what are we going to do with all this information? But anyway, the information keeps a lot of people employed, looking at screens and things like this. But you also have the observer effect, which is the Heisenberg uncertainty principle applied in human tissue. By looking at it and disturbing it, you have changed it. So what she's looking at may no longer be what's happening inside your breast or colon or wherever they're taking the tumor from. Now, again, we already heard about Ioannidis and his papers before. This is a paper from his group showing that the new cancer drugs aren't working. And the bottom line is they looked at 92 drugs from 2000 to 2016. And what's very interesting about these therapies, if you look at the tumors of the patients that were treated with these various kinds of precision drugs or whatever you want to call them, it looked like the tumors were, wow, the tumor's really responding well. It's unbelievable. But the people don't live in it very little, like 2.5 months longer. What I understand is that the Food and Drug Administration determines whether a new drug should be given to human beings based on progression-free survival. That means, what does the drug look like it's doing to the tumor? And if there is some modicum of overall survival benefit, well, that's great. But we really should be looking at, does the drug keep you alive far longer than any of these other drugs? And they don't look at that. That's not part of what they're looking at. So now, evidence challenging somatic mutation theory. And whenever you challenge the dogma, you end up like Bruno, right? So you got to be careful. So people don't want to look at it. They don't want to talk about it. They don't want to hear about it. And then, of course, we always like to Einstein. We're all big on whatever Einstein had to say. No amount of experimentation can prove me right, but a single experiment can prove me wrong. Well, let me show you stuff that should have proved the somatic mutation theory hopelessly wrong, okay? And why we continue to think that cancer is a genetic disease. And I'll let you guys make the decision. So Theodore Boveri, in 1914, wrote this paper, a purely speculative paper, where he thought that cancer might have something to do with abnormal chromosomal behavior, movement of chromosomes. He came to that revelation looking at sea urchins. And he even said, I have no clue how sea urchins are related to cancer. And even his friends in cancer said, Theodore, I think you're out to lunch. Well, he said, well, I'm probably totally wrong, and I want to apologize to the entire cancer community that I'm wrong. I have no knowledge. I'm ignorant. And now we've anointed him as the father of the somatic mutation theories. So go figure. Now we're looking at, now this is really interesting. We're finding some cancers that have no somatic mutations. Greenman showed this, Baker, Parsons, and those are big papers. So you say, wow, look at that. There's a bunch of he found 30% of breast tumors using a very powerful genomic sequencing analysis had no mutations. Yet some other breast cancers had thousands and thousands of mutations. But I was looking for how you explain that in your somatic mutation theory. Nothing. Same with Parsons. They couldn't find in one of the GBM papers, patients had no mutations in any of the major signaling cascades. Oh, interesting. Now, new data are showing us that these cancer driver genes are abundant in all of our cells that never become cancer. It's unbelievable. So if we took samples from we'd have loaded with driver genes. You go down to Dana-Farber and they say, oh, we're going to start radiating poison. You got all these mutations. So what's wrong with me? I'm perfectly healthy. Oh, no, you got the driver mutation. So, and this can also account in part for when you give immunotherapies to some people. The immunotherapy sometimes kills you before the cancer does. The problem is, of course, and Martin Carina pointed that out. Okay, we're going to target precise, there's that epitope. So, oh, my kidney also has that. Well, we're going to take your kidney out along with the cancer. What's going to kill you faster? So it's called hyper-progressive disease, and it's a well-known phenomenon in cancer. Some cancers don't cause mutations, like asbestos, right? Asbestos. The rarity of cancer it's rare in Aboriginal tribes. Okay, people who live according to their traditional ways, like these Africans and Inuits from the Arctic and all these. The British were shocked. Albert Schweitzer couldn't figure out. Well, he looked at 10,000 Africans through these. Nobody had cancer. It was strikingly different from the Europeans. And the Inuits, I went to the medical school at Thunder Bay. I was invited there to give a talk some years ago. It's a medical school in Thunder Bay, Canada, and they service the Inuits. And they're massively unhealthy. Obesity, diabetes, dementia, all kinds of things. And yet 100 years ago, they were some of the healthiest people on the planet, right? And all they did was eat meat and fat and stuff like this. They weren't eating vegetables or fruits or very many, all except seasonal. Very interesting. When a Western diet lifestyle enters your population, brace yourselves. You're going to have all kinds of chronic diseases. And then, of course, our closest biological relative, the chimpanzee, at 98% similar to us in gene and protein sequence. And there's never been a documented case of breast cancer in a female chimpanzee. And breast cancer is causing a big problem in females and even males with this disorder. What's the difference here? The chimp is living according to his diet and lifestyle, whether he's in the forest or whether he's in Franklin Zoo down here in Boston, whether he's in San Diego Zoo. I was at the San Diego Zoo and I said and I was also down here, I talked to the vet. I says, why don't you give the gorillas go out and get a big box of jelly donuts for these things? I'm sure they would like it. Oh yeah, they really like it, but it's very unhealthy. I said, what the hell? And I'm standing here eating the jelly donut, right? I start looking back and what's going on. It's just that we're not allowed to do these kinds of experiments on a chimp. It's called animal cruelty. Can you believe it? So I checked. Go down for yourself. Ask the vet. Now the nail in the coffin came from what I simply did was went out and looked at all these studies that transplanted nucleus from tumor cells, moved move both nucleus and mitochondria back and forth between different cells. I didn't do these experiments. What I did for the very first time is reinterpret it again, reinterpret the data that's already there in a different light. And I published this paper. This was actually, I did it in my book in 2012, and this was 2015. It's got over 100,000 views and downloads right now. And this is the very simplistic summary of dozens and dozens of replicated experiments. Replicated. So green cells get other green cells. Normal cells beget normal cells. The nucleus is healthy. The mitochondria, number, structure, and function are healthy, and they produce cells that are regulated in their growth during normal turnover and things like this. Red cells are tumor cells. Tumor cells beget tumor cells. So the tumor cell, of course, for the majority of them, they have genetic defects. That is true. Some don't, but many do. And they also have abnormalities in number, structure, and function of the mitochondria. And what is causing the dysregulated growth? Is it the mutations in the nucleus, or is it something in the cytoplasm, abnormalities in the number, structure, and function of mitochondria? So when the red nucleus from the red cell is placed in the green cytoplasm, this was done by Israel and Schaefer, in vitro and in vivo both, they got regulated growth, which was scratching their head. And then they took the nucleus from the normal cell and put it in the cytoplasm of the tumor cell, and they got dysregulated growth. This is just the opposite, just the absolute opposite of what you would have expected if driver genes are controlling dysregulated cell growth. You wouldn't get these kinds of experiments. Newer experiments now, if you take the green mitochondria and purify them and put them into the red cytoplasm, you get regulated growth. And if you take abnormal mitochondria and put them into indolent cells, they become explosive. So clearly, what the nature is telling us is that this is a disorder driven by mitochondrial dysfunction, not by mutations in the nucleus. So if somatic mutations are not the origin of cancer, then how do cancer cells arise? We need to know the answer to this. And this goes back to the work of Otto Warburg, the German scientist, a fascinating book written by Sam Appel. He was a Jewish scientist who Hitler spared. Hitler feared cancer, kept Warburg alive during the Holocaust with the hope that he would cure cancer someday. But what Warburg actually found is that cancer arises from chronic damage to cellular respiration. We're breathing. We're all breathing here. We're getting our oxygen from oxidative phosphorylation. Energy through, this is what we're working on, substrate level phosphorylation is a non-oxidative form of energy. You move a phosphate group from an organic molecule onto ADP to get ATP. It's different from oxidative phosphorylation. It's an ancient pathway. These are the way cells got energy before oxygen came into the atmosphere 2.5 billion years ago. So the cancer cells upregulate these ancient substrate level phosphorylation pathways. Cancer cells continue to ferment glucose in the presence of oxygen. Aerobic fermentation should not happen. It's called Warburg effect. We're working on that, all the confusion about Warburg's effect. Also, the red is our big contribution to the cancer field. We now have found that amino acid glutamine, the most abundant amino acid in our bloodstream, can also be fermented in cancer cells. This is different than what everybody thought. They thought it was respired. Derek Lee sitting here has got some of the strongest evidence, and I'll be showing you that. We work with Christos Xenopoulos at Semmelweis University in Budapest, Hungary. He's the world leader on substrate level phosphorylation in the mitochondria, and we're now doing research. So Warburg didn't know about this, okay? So they threw Warburg under the bus. When Watson and Crick discovered the genes, everybody ran, oh, these tumor cells are loaded with mutation. What happened to Warburg? He's passé. He was right. Essentially right. He just didn't have all the parts of the puzzle. We now put some of those parts out, connected the dots, and shown he was essentially correct. So enhanced fermentation is the signature malady of all cancer cells. Now, if you look at a tumor and you go down and get the woman to look at all the different mutations, every cell in that tumor has a different constellation of genetic mutations. They're not all the same. This breast tumor, that breast tumor, they're all different from each other. Nobody has the same. Even within the same tumor, all the cells are different from each other. But one thing is common to all of those cells. They are fermenting. They're getting their energy from a non-oxidative source. So the key to manage cancer is to take away the fermentation fuels, okay? I mean, not complicated, and you're going to come to realize that. So what's the evidence? Are you sure about that? Well, let's look under the electron microscope, because that's when you can really see mitochondria structure, and you can isolate them and look at their function. So the cell in those nice, beautiful stripes, they are the cristae. They contain the proteins and the lipids that drive the electron transport chain. Chris showed a picture of the electron transport chain. So that's a healthy mitochondria. The one on the right, that's a glioblastoma mitochondria, deadly brain cancer. And you can see the cristae are missing. It's called cristallysis, a ghost mitochondria. Structure determines function. It's an evolutionary conserved concept. If the structure of the organelle is abnormal, the function of that organelle will be abnormal. So you can see that. And now what about breast cancer? On the right, the HMSC, there's normal breast cells, normal breast cells, the nice, beautiful stripes of the cristae. Don't forget, we brute forced these mitochondria. We actually took out all the lipids. We analyzed everything in incredible molecular detail. And the cancer cells have very abnormal lipidome and you can see the vacuoles in the breast cancer. Those spots, there's a vacuoles. Colorectal cancer, CRC, mitochondria that are empty with cristae. Structure determines function. Now what I did is I went back and looked at all human cancers that we have studied over the years, decades, from the 60s, 70s, 80s. In the early days of the electron microscopy development, people were looking at everything, and they were recording what they saw. So I just went back and said, look, these guys are seeing all these different cancers, abnormal structure and function, appearance of mitochondria. It represents over 90% bladder, breasts, colorectal, gliomas, blood cancers, livers, melanomas, osteosarcomas, pancreas, prostate. They're all similar. They all have a problem. They have abnormalities in number, structure, and function of the mitochondria. In our most recent study, with Dr. Ta here, we put that together. When you can't respire, lipids can be deadly because they'll create reactive oxygen species and potentially kill the cell. So what the cell does is it vacuolates them. They put the lipids in these lipid drops, LD, lipid drops. That's a sign that the mitochondria aren't working, because if they were used, those fatty acids, they'd explode from the ROS. So to protect the cell from death, they store them in these droplets. And you can see, we went through, Lynn and I, all the major cancers. All of these are different: colorectal, breast cancer, blood cancers. They're all storing lipid drops. It's a marker for deficient oxidative phosphorylation, meaning that those cells need to ferment. What do they ferment? Glucose and glutamine, right? How come nobody's targeting the glucose and glutamine? Too simple, very simple. Can't do simple things. Okay, so when we look at energy metabolism in normal cells, here's a very simple, the glycolysis is called the Embden-Meyerhoff-Parnes pathway, documented well. Established glucose is metabolized through a 10-step process to pyruvate. Pyruvate goes into the Krebs cycle, fully oxidized, reducing equivalents, these NADH, FADH2, deliver their electrons to the electron transport chain, and lo and behold, we get tremendous amounts of energy, and with the waste products of CO2 and water. CO2 and water come right out. But we also get a little energy from substrate level phosphorylation. These are the ancient pathways that existed for energy metabolism before oxygen came into the atmosphere. So we have a little bit of energy in normal cells from those pathways. But the cancer cell, if you notice it, you see the shift. You get a lot of energy now from these ancient fermentation pathways called substrate level phosphorylation. And you can see 40, we're not really sure. This is one of the debating points where we're not sure. All we know is that energy through oxidative phosphorylation is neither necessary nor sufficient for driving dysregulated cell growth. Glucose and glutamine fermentation in the cytosol and the mitochondria, mitochondrial substrate level phosphorylation, are necessary and sufficient for driving dysregulated cell growth. Now, I don't want to bore you with these. We love these things. We can spend hours looking at this stuff. But you can see glycolysis and glutaminolysis. Those are the two driving pathways that provide the energy for dysregulated cell growth and the metabolites needed for new growing cells. And you can see these star bursts here. So glutamine, glutaminolysis, glycolysis, and energy. These cancer cells are in fact taking in oxygen, but they're blowing out reactive oxygen species that are carcinogenic and mutagenic. So we're not getting very much energy from oxphos. We're getting more energy from substrate level phosphorylation with succinic acid and lactic acid as waste products, not CO2 and water, succinic acid and lactic acid, and we've measured that. Derek's measured that. Now this is Derek's gift to mankind. He put a lot of energy. Derek, stand up there. Well, just let people see who you are. There he is, right there. So he put a lot of energy into this, and we thought about how we're driving dysregulated cell growth through using glucose and glutamine as fermentable fuels. And Derek came up with the idea that this is a high throughput. These cancer cells are sucking down these two fuels in enormous amounts relative to normal cells. And we're blowing out lactic acid and succinic acid into the microenvironment. You know, one of the biggest things in oncology is my immunotherapies don't work, my chemo doesn't work, my radiation doesn't work. There's so much acidification in the microenvironment, it's blocking all these things from working. Where's it coming from? Glucose and glutamine. If I take away glucose and glutamine, these things now become vulnerable. But nobody's doing that, right? Nobody's doing that. But anyway, Derek showed that all of the linkages, these two powerful pathways, are feeding off of each other in a synergistic way, driving the dysregulated cell growth and the metabolites needed to make new cancer cells. And then we put this together. Okay, we got the beautiful normal mitochondria on the left, and over time, chronic disruption of oxidative phosphorylation leads to ghost mitochondria. The delta G prime ATP, this is the key that you have to know that is underlying the energy efficiency. So we're keeping the energy efficiency, but we're shifting. The green line goes down, that's oxidative phosphorylation, and then there's a protracted increase in substrate level phosphorylation. To bring Einstein back again, substrate level phosphorylation is linked to malignancy as strong as gravity is linked to the redshift. Anybody know what Einstein's redshift is? The pull of gravity on photons, okay? It was an unbelievable thing. That's how strong the linkage is. So we can now put all the parts of the puzzle together. Again, the common pathophysiological mechanism for cancer is damage to a mitochondria that can come from radiation, chemical carcinogens, intermittent hypoxia, systemic inflammation, rare inherited mutations, RAS oncogene, oncogenic viruses, older age. Every one of these things impacts negatively on the structure and function of the mitochondria, producing reactive oxygen species, ROS, which are carcinogenic and mutagenic. So most of the mutations that we see in the nucleus of the tumor cell are downstream effects of mitochondrial damage and dysfunction. Yet we're chasing tails, we're not chasing the origins. So Ross, go on. Now the oncogenes turn on and they're floodgates. Those are those high throughput pathways, or glycolysis and glutaminolysis are driven because the mitochondria are suffering. So how are the cell going to survive? It has to get an alternative energy. The alternative energy is substrate level phosphorylation, driven by glucose and glutamine. And now we can take all of the hallmarks of cancer produced by Robert Weinberg and Hanahan, and link every one of those back to dysfunctional, failing function of mitochondria. Now, there's a book, Sid Mukherjee wrote the New York Times Pulitzer Prize winning book, The Emperor of All Maladies, all right? Mukherjee. It was on the bestseller list for a long time. He struggles in the book. I don't understand. What's the common pathophysiological? I don't understand. I don't understand. And the end of the book is very bleak. Yeah. It's true, read it. I said, for Christ's sake, did he read he should have read some of our papers. He wouldn't have been, he might have the depression that you have to treat with it. He should be on a ketogenic diet or something. Jesus. So now, if most cancer cells obtain energy through fermentation, what therapies might be effective for managing it? Well, one approach is let's target those, reduce those fermentable fuels. And now I'm going to show you the evidence. I'm going to show you the power of predictability, as Greg would say. If you're on the right path, you should be able to predict something, right? So let's see if we can predict. So here's the strategy: metabolic management of cancer following changes in plasma glucose and ketones. Okay, let's lower the glucose, water-only fasting. I'm going to talk about that in a minute. Calorie restriction, restricted ketogenic diets, and then raise ketone levels, because it's the glucose that's driving the dysregulated growth. And now we're going to give them fatty acids and ketones to choke on, right? And not only that, it'll make the rest of your cells healthy. We're going to increase the delta G prime ATP hydrolysis. This is the key for ketones are effective because they increase your delta G prime. Richard Veach, good friend of mine. Now look at this. The ad libitum is to give the mouse all he likes. Let him eat as much as he wants. And then the other guy, the CR group, calorie restricted, 40% calorie restriction, 3 days post-inoculum. And you can see we got a huge reduction in the size of that tumor. It's obvious. people say, wow, all I have to do is cut my calories back by 40%? Well, that's pretty impressive. Nah, wrong. Okay, 40% in a mouse is water-only fasting. Do you have another, there's something else you can do? You know, they don't want to water-only fast. You can't get people lining up for that, right? But in any event, this mouse have no choice. We put them in a cage, we just give them what they amount. So now here's something I want to share with you for Greg's amusement. He doesn't know about this. Anyway, I don't know, when he first saw my book, what does he do? He turns it over to his father to ask if I'm right. Right? He suffered having to measure nail lengths as a punishment when he was a kid. So what he did to me, he's punished me by giving his father my book and having him eviscerate my data. So all these red lines are Jeff Glassman's critique of what, so I had to sit down and spend days and days paging out exactly what we did, how many mice. I gave him the raw data from the experiments, and I let him look at it, and I explained every one of his little red lines there and what he was concerned about and all this kind of stuff. And I did, Perna says to me, my other staff member, boy, this is a smart guy, but we're not that dumb. So we had to spend our time going back to address every one of Jeff's criticisms. And finally, he writes me a letter, but maybe more satisfactory to me. Thank you very much for explaining. I actually accept what you're saying. Okay, I got it better than you.
He actually thought you understated your case mathematically.
Yeah, well, he might be right about that, but I wasn't understating. I had to present the way we looked at it. But it was a great experience and exercise for us to look at how someone else's brain would view the information, and if we were able to communicate the information back and forth to satisfy both groups.
Yeah, you know, regardless of what he said, I was kind of done with it, the nuclear and mitochondrial transferase experiments.
Yeah.
That's enough.
Yeah, well, but for a lot of people it wasn't. But you tell me. You're the guy with all the answers, right? No, I don't know either. It's called confirmation bias, dogma. Dogma. You can't change religion in people's brains.
For a more elegant experiment to demonstrate the case compelling.
Yeah, well, I put everything out there, and people still say cancer is a genetic. I mean, what are you going to do? So, but anyway, we got a lot of work from Jeff's work, and it's when you have people that are not directly in your field, and they see the data, and you have to examine how they are viewing this information. Therefore, our explanations need to be more clear and accurate to make others also recognize what we're doing. But I thought you'd enjoy that because we had to spend a lot of time addressing those issues. But in any event, predictability, which is very important. And each square is a mouse under a different dietary condition. And we did linear regression analysis, which is actually choosing one as the causative and another one as the effect. So glucose was the independent variable. Beta hydroxybutyrate is the ketone body in the blood, and the tumor weight is the size. So what we see here on the left is that blood sugar goes down in these animals, ketones go up. This is a evolutionary conservation of energy. When you stop eating, if the brain doesn't get energy, you're going to go unconscious. So the body mobilizes fat, turns them into ketone bodies, water-soluble fat breakdown products, and that's clearly related to low insulin, higher glucagon, more ketones. But on the right, you'll see as the glucose goes down, the size of the tumor goes down. The tumors got smaller as the blood sugar went down. And the higher the blood sugar, the faster the tumor would grow. This is the first evidence in the scientific literature that I'm aware of predicting that rate of tumor growth is linked directly to blood sugar. Since this has been pointed out, we've seen it in glioblastoma, colon cancer, breast cancer, bladder cancer, all the different cancers that have been looked at. The higher the blood sugar, the less likely, or the earlier the patient will die. So there is definitely a predictability in cancer for blood glucose levels. Now what we did, based on a lot of interaction with cancer patients, we built the glucose ketone index calculator, which is a quantitative assessment of the ratio of glucose to ketones. So glucose is usually measured in deciliter. We convert that to millimolar, and then we measure ketones. So you have this GKI. And what we've determined that levels of 2.0 or below put one in nutritional ketosis. And what is nutritional ketosis? It's essentially the way we existed during the Paleolithic period. We had a lot of exercise, a tremendous amount of exercise. We had no highly processed carbohydrates in our diet. So we had to move a lot. We ate just predominantly low carbohydrate plants and animals, mostly animals. What we're doing is simply taking people, and people say, oh, what do I have to eat to get a GCAI? Yeah, you can do it with a ketogenic, you can do it with a Mediterranean diet, you can do it with a pescatarian diet, we can do it with any kind of a diet. And each person would have to adjust for themselves, whether it's cultural, religious, or whatever, to get their GKI. And now we're fine. I built this for the GBM patients, and we switched to all cancer patients that will benefit from this. But now, as a prevention of cancer, healthy guys like these CrossFit people and these other people, they want to see how low they can get their GKI because they're back in the Paleolithic period. My good friend Dominic D'Agostino lives in this environment constantly. So it's a very good way to prevent cancer as well as managing it. Now, what we did here was we took, Press Pulse comes from Ahrens and West, paleobiologists for destruction of exterminations of plants and animals on the planet. But we actually adopted the concepts. We have press therapies and pulse therapies. The press therapy, you put them on restricted ketogenic diets, water-only fasting, this kind of thing, even ketone supplements. Stress management, like Chris was saying, very, very important. Because when you're under stress, glucocorticoids go up, all right? So you got to keep your glucocorticoids down. You got to reduce stress. A lot of people with cancer are freaked out. They all think they're going to die. So stress management is very important, lowers the glucose, makes the metabolic therapy work better. Then you come in with dosage, timing, and scheduling of glucose targeting, glutamine targeting, and hyperbaric oxygen, which can eventually replace radiation for killing. You can kill tumor cells by reactive oxygen species, but you're enhancing the health and vitality of the normal cells while you're selectively destroying the tumor cells. So we move the patient from disease state to a managed state. And then with improvements in dosage, timing, and scheduling, we can move from the management potentially to the state of resolution. Dr. Duryea, also working in my lab, we have a major paper under review right now from a major medical journal with more than 20 MDs and PhDs on this for the metabolic management of glioblastoma, which this is the framework, but the actual nuts and bolts of how to treat cancer patients on a day-to-day basis and all the things you might encounter in trying to treat individuals, personalized nutrition, all of these kinds of things is in that paper. So when that paper comes out, there's no excuse for anybody not to treat cancer patients because here's the how-to manual to do it. This was the framework for doing it. So let me talk just a little bit about glioblastoma, terrible brain cancer. This is what killed John McCain, Ted Kennedy, and many other folks, Bo Biden, the president's son. And it's a terrible disease. You can see how bad the brain looks. The brain gets, patients die from intracranial pressure. Those purple cells on the right, those are the tumor cells. They go across the surface of the blood vessels called Virchow-Robin spaces, and they can see the entire brain. Even though you can't see the tumor cells, you see only this discoloration and cystic area, but the tumor cells are out. So that's why they always say we can never get all the cancer. It's always there to some extent because the tumor cells have already seeded out into the parenchyma. Now, here's another very interesting thing. Let me just brace, I just have predictability. What do you call, failure to replicate. Emily, didn't you say that? Greg says failure to replicate, right? Okay. Nothing is more replicable than how fast you're going to die with the GBM. Look at these. Look at this. This is 5 surgical institutions. You can't design more replicable survival data than this. All these, and we can go all over the world. In every major brain cancer center, you're going to get a survival curve that looks like that. Highly replicable. We just heard about how hard it is in the oncology field to replicate data. Look how replicable this is. And there hasn't been any improvement in 100 years. Right? 1926, 8 to 14 months. 2024, 8 to 16 months, median survival. I said, what's going on? 100 years, we got Webb telescope for crying out loud. And we can't move the needle on this GBM. Why? What is going on for why we're not moving the needle and survivability for this cancer? And we published this paper, and you asked me why people don't listen to what we're saying. When you debulk the tumor, cut it out in mass, you create a wound. And then as soon as the patient wakes up, you start irradiating the patient, right? That breaks apart the glutamine-glutamate cycle in the brain, freeing up massive amounts of glutamine, the fuel that's one of the two fuels. And when your brain is irradiated, they give you high-dose steroids to reduce the inflammation, which gives you hyperglycemia. I just showed you, the higher the glucose, the faster the tumor is going to grow, right? I mean, it's obvious. The two fuels driving the dysregulated growth are created by the standard of care. What we're doing to these poor people is denying them, for the most part. You saw how replicable. It's because everybody on the planet is getting the same treatment. There's no differences. Standard of care happens in India, Germany, Japan, all over the world. They're doing the same thing. So this is explainable. It's explainable because you're not allowing the person's brain to recover from the surgery and targeting the glucose and the glutamine simultaneously. So we developed the therapy using the VM3 GBM mouse model, which has all the growth characteristics as I just see in human GBM, all the ways they grow. And we use this drug DON, D-O-N, deoxyoxynoreleucine, which is a glutamine analog. And the glutamine analog looks like glutamine, and it blocks glutamine utilization. So we put the mice on a ketogenic diet, lowering the blood sugar, elevating the ketones. And this is we have tremendous data. I could spend hours with you. But overall, what's the success is survival. Survival is success. And what you get is the blue line, they're dying fast. The green line, the diet by itself. The red line, the drug by itself. And the purple line is when you combine the diet with the drug. You can see how powerful it is. We tried this on a Egyptian guy. He did really well at the beginning, but they insisted on him being irradiated. I said to Ahmed El-Saka, the attending physician, I said, the guy's doing great. He's back in the field. He's a corn farmer. He's a young guy. Oh no, we got to irradiate him. I said, Jesus. So anyway, I said, oh, well, I'm here in Boston. He's there in Alexandria, Egypt. So anyway, the guy, I said, how's the pay? Yeah, he seems to be doing okay. We published the paper. Then at 30 months of age, he starts getting headaches. And he dies at 30 months of age. And when they did the autopsy, he died from the liquefaction of radiation poisoning to the brain. His brain was liquefied by the very treatment that they used for him. It was very disheartening to me. Then I also wrote this paper, provocative question. Harold Varmus runs the NCI at the time, Nobel Prize guy. So we had provocative question. They wanted the provocative question, change the standard of care for GBM. Oh, can't do that, right? So anyway, we're not going to drop the needle. We're not going to move the needle until we stop irradiating the brains of these poor GBM patients. And we say that in the big treatment there. But radiation brings huge amounts of revenue. Follow the money. So once we have this, Thomas's protocol, the one we write, is going to have a major impact if we can stop irradiating these poor folks. And I always show this one, poor Brittany Maynard, remembering Brittany. She was a young woman, January 2014. Here she is with her husband right after she got married. She gets a glioblastoma, actually from a lower grade tumor that exploded into a GBM. She puts her paper, People magazine, I'm going to kill myself rather than take the standard of care. But you can see her face is swollen. That's moon face from high-dose steroids. When you give people high-dose steroids, they get this thing called moon face, and you can see it there. So she dies with dignity with her family November 1st, 2014. Now this is our man Pablo. We wrote a case report on him. He came, same year as Brittany, glioblastoma, no radiation, no chemo, no steroid. He didn't want any of that. The attending physicians said, you're going to be dead, Pablo, in 9 months. Start getting your affairs in order. He said, to hell with it, he wanted to have kids. He says, if I take radiation and all that stuff, I'm not going to have kids, much less I'm not going to live to have kids. But in any event, he rejects that. And I'm going to talk about him in a minute, survive with good quality of life until just this month. But in any event, we look at his brain tumor. You can see it when it was this big thing with the arrow. Then at 2016, you can see it's bigger. So we had his first debulking surgery in early 2017, cut it out, and we had 5 years of Pablo Kelly measuring every day his glucose ketone index, his glucose and ketones and the index, and we plotted it to let other people know what you might want to do to reduce the rate of growth. His tumor was cut out again, grows for another 3 years, cut out again. In the meantime, he gets married. He has 1 child. He has two children. And we found out that in addition, he had the IDH1 mutation, which actually inhibits the glycolysis and glutaminolysis pathways, giving him a survival advantage, slight. And I don't have time to talk about that, but if anybody's interested, I have the crew here that can go into a deep dive on that. But the idea is Pablo did have an advantage. He used a special diet and lifestyle together with a God's gift of a therapeutic mutation. Not all mutations are bad. Now here's Pablo. Thomas and I had a discussion with him in August. We were laughing about Pablo. We were laughing because he survived 10 years. He had survived 4 operations on a previously defined inoperable tumor. So we had a big laugh about this. Pablo, how many times, what are you going to, you're going to outlive me. You're going to have 20 operations on your head and all this kind of stuff. We were laughing. He was perfectly healthy. There was nothing wrong with Pablo. And then 2 days later after that, to our surprise, they say, oh, we want to go in again. There's some residual. We think we can get it out. So he comes out of the operating room right here, thumbs up, talking and everything. And then 12 hours later, cerebral hemorrhage and dead from the operation, not from the tumor. He was our poster child for how long you can live with a glioblastoma. Everybody was looking at him, coming for hope and everything. The poor guy. Can you believe this? And I know poor Danny from Marshfield, Massachusetts, right down here on the coast, diagnosed in 2017 with pineoblastoma, goes through the spinal cord and everything. You can see his fat face from all the steroids, radiation, everything they gave this poor little kid. And then he died in 2021 from standard of care. Now, because of that, we got a grant from British UK Childhood Center to study childhood cancer, thankfully for them. And we developed a pediatric high-grade glioma model in our lab, Purna Mukherjee and myself, where we can put tumors into the brains of young mice at the same age as Danny Sheen and little kids. And we could see the tumors going down the spinal cord. So we replicated key aspects of pediatric oncology in these mice. Now, I don't know if I can, yeah, maybe I can get it. So, so you can see the difference. So the guys on the left are the guys that were not treated with metabolic therapy, and they were going to be one died already. But you can see the guys on the right, we gave them ketogenic diet and bendazole and Dawn, targeting glucose and glutamine simultaneously while raising ketone levels. And we're keeping these guys alive so much. This is going to be a blockbuster in the pediatric clinics. But they don't use it. Because cancer is a genetic disease, haven't you heard? It's unbelievable. This woman here was treated by, I'm on the paper too, Ikasiki of this, a clinic in Istanbul, Turkey. And you can see she was from Ohio, and she had breast cancer to the brain and many, many other organs. So all those dark spots are the spread of the breast cancer around. They gave her metabolically supported chemo for managing NSAID. They said she had 1 month. 1 month to live, get your affairs in order. Radiation, chemo, did all these horrible things to her. So there was nothing working. So she jumps on an airplane, goes to Istanbul, almost dies in the ICU for 2.5 weeks, according to Abdel Slocum told me. Then they put her on this metabolic supported, low doses of chemo, very low doses. We're not throwing out, like Chris said, we're not throwing everything out. There's some things that can work when you do it the right way. Anyway, here she is celebrating her life with her husband in Hawaii in 2021. And this last week I had to call because I know people are going to ask me what's going on with the woman. So I called up Slocum. I says, Abdel, how's this woman doing? I don't want to. Oh no, she's fine. She's got a nice letter thanking him for all the wonderful life she has. She was a goner. She's doing fine. We got this guy from Greek. He had lung cancer to the brain, restricted ketogenic diet, primary lung cancer, amyloidosis guy. He's doing fine. He's out 10 years. I wrote a puff piece for Nature on how we can manage prostate cancer. I did all these things. And then, of course, with the dogs, Jimmy's here, his dog with the blueberry tumor on his face. This woman came to me, published this, right? I had to rake up some veterinarian to make it look like Lauren Nation's a friend of mine. I said, Lauren, come on, join us, because I'm not a vet. So, and the fact the woman treated her own dog. This was a mast cell tumor in the dog's nose. And the woman, first thing she does, okay, no more carbohydrates for you. Poor dog. It's a pit bull. So what happens is that it shrunk down, raw egg, raw chicken, fish oil, and 40% calorie restriction, just like the mice. And lo and behold, the tumor disappears. Here's the dog in 2013. In 2016, complete. They say, oh, you got to have radiation chemo. And it's going to cost a lot of money. Dog could be very sick, diarrhea, all kinds of stuff. I said, no, I don't want that. So no surgery, no radiation, no chemo. And look at that. The dog died at 15 and a half years of age from heart failure. So it didn't die from the therapy. In fact, the therapy kept the dog alive even longer, got rid of the face. And don't forget, I published this autolytic cannibalism. Autophagy, like Chris was saying, where you rearrange mitochondria and organelles within the cell. But there's also autolytic cannibalism. When you put the body under nutritional stress, every cell in your body has to justify its existence to function as the whole. You got a tumor, those cells aren't functioning well. Eat them. So the body will turn on them and attack and eat the tumor for the good of the survival of the whole. So we have stage 4 cancers. We don't consider terminal cancer anymore. This is not right. If you do metabolic therapy, we don't know whether you're going to die or not. Cancer Revolution by Maggie and Brad Jones. Maggie is a long-term survivor of lung cancer to the brain, and she is collecting all of these folks, okay? A registry of all these people that are alive that should have been dead. And she's got dozens of, I'm alive. And she's got all these people that are doing various, we haven't even standardized this yet, folks. Once we standardize this, we're going to drop the death rate on this cancer by 50% in 10 to 15 years. The problem is how you get through the wall of obstacles that say you can't do this. Everything is you can't. Remember that guy telling us you can't do that? Who the hell are you? These people need to live, for Christ's sake. And then we look at this, and yes, mitochondrial metabolic theory can explain better the origin and management of cancer than the somatic mutation. When are we going to get rid of this noose on our neck with the somatic mutation crap? Okay, we can keep people alive, and yeah, in our conclusions, yeah, it's not a genetic disease; it's a mitochondrial metabolic disease driven by substrate level phosphorylation. And not only that, I put at the very end here, press pulse ketogenic chronic diseases. We're right about this chronic disease. I mean, it's all related to the lack of exercise, too much carbohydrates. Get a GKI, everybody got a 2.0 GKI. The hospitals be running down the streets looking for somebody to treat. Everybody would be like a CrossFitter, for crying out loud. They're all going to be healthy, but we have a picture in the lab. It says pills and surgery, and you have 100 obese people all lined up trying to get the pills. Diet and lifestyle change, not one guy in the line. Right? So we're up against a tough sell. We're having a global society of cancer metabolic therapies. We're collecting collaborators from all over the world. We want to set up clinics. We have people talking to us like, okay, once the paper comes out, how are we going to set up clinics? What are we going to do? What's the nuts and bolts on this whole thing? And of course, we have to thank so much our support, Greg and Broken Science. And yes, CrossFit, when Greg ran it, that was definitely helpful to us. Broken Science, Cork and Family for sure, philanthropy from Maroon and Edward Miller, Kenneth Raining, Children with Cancer, United Kingdom. Our university has been helping us tremendous. Delaware County, a Deputy's Benevolence Fund. Oh yes, I can support them. And in the past, the National Cancer, the NIH, National Institute of Health. So I will thank you for your attention, and I'll be happy to answer any questions if there are any. Thank you.